Welcome, everybody. We're going to do the 3D neuron analysis for the Avia Online Workshop for today. So I'm just going to do a few recap of things concerning Avia. It'll be really brief. On this slide, I'm basically wanting to kind of capture all of the development that we've been doing since the start of Avia. So Avia itself has only been a product for DR Vision in 2017, and since then we've had a very aggressive development pipeline. The newest version, Avia 9, I'm sure you have heard it from Jim and Trevor and everybody on the team here, has to deal with cell and multi-scale analysis. This is a cell that we analyze, not only the cell, but the nucleus and all of the organelles, and you can scale that to kind of the entire organism also. So that was the big thing that we just launched. It's been a week, I think, exactly a week since we launched this. So just a lot of development happening, and that will be continued as for this year and the foreseeable future too. And then just a brief look at the Avia team here. This includes the devs and all of the managers that are involved in developing in Avia, and also the Q&A team too. So lots of efforts and lots of collaboration happening here for the development of Avia. And to mention collaborating, we also have a number of academic collaborators, and they really are one of our driving forces in the types of things that we add and how we add them. And also they help us organize how well these features fit and work within the kind of the scientific fields and things like that. So they help kind of navigate us to the features and things that we develop here at DR Vision. All right, so that's just a brief introduction to Avia and kind of the team, and I'll just launch right into the Neuron stuff because I want to focus that for the workshop here. So just to kind of give you a brief look at our mindset when we're generating all of these 3D neuron tools, and overall for Avia is we're trying to tackle some of the most common tracing issues that you see for neuron reconstruction. So one of the biggest one is this, because they're so complex and some of these data can be very large, one of the things we wanted to tackle was the slow processing of some of these data sets. And we'll talk about this in more details, but we implemented what's called a voxel scooping algorithm to address some of these concerns. The other piece we're looking to address is the uneven staining that can occur throughout the dendrite. This can cause, especially for some of the automatic tools, a lot of headaches and issues when you're trying to trace a complete dendrite from end to end. And we're tackling that with an algorithm called adaptive thresholding. I'll go over that in the next few slides very quickly for you too. And then, of course, some of these data sets, because they're large, they can also be complex, and that makes it very tricky to get the right tracing and to connect all of the tracing correctly. So with this, we've implemented ROI processing, so you can basically focus on the areas that you want to, or smart cropping, which I'll show you, and it kind of comes into play when we're looking at some of our more manual tools. And that's just to kind of help decrease the complexity when you're working in these tools and in these data sets. Okay, so first what we're going to talk about is the automatic 3D neuron tracing. I'll be talking about more of the manual piece in the next section, but we'll talk about the automatic piece first. So basically, like I said, one of the algorithms we implement is the voxel scooping algorithm. And this is, again, to address kind of the slowness of this. So how I like to describe this is kind of imagine ice cream scooping, essentially. And so what the algorithm is doing is it's scooping a set of voxels, and it's taking a look at that set of voxels and determining, based on the parameters that you input, the thresholds and whether it meets the threshold of that being a dendrite or not a dendrite. And if it is a dendrite, it's going to scoop the next set of voxels and basically ask the same question. is this within the parameters of the dendrite? If it is, it'll continue to scoop along that direction. However, if you get to a point where you're scooping basically a set of voxels that is not a dendrite, it doesn't pull that into the dendrite, and it also stops scooping in that direction. So basically what that means is it's only really processing the data that is that is part of the dendrite. It's basically eliminating or not even processing the background images. So it makes it a lot faster in terms of the tracing of the dendrites. I also want to just briefly say, if you have any questions or if I'm going too fast, please let me know. You can ask your question in the chat or just kind of speak up, and I'll answer whatever questions that you have. All right. So the next piece is adaptive thresholding. Basically, this allows Avia to be somewhat flexible with where it determines the background levels are. So if we kind of take a look at this simple example I have here, where the top right of the original image is significantly brighter than the bottom left of this Sudoku image. You can see that if we did a global thresholding, you'll be able to only partially get the image, and everything else would probably be black, or you're gathering too much information and it will just take a significantly longer period of time. So what we do is doing the adaptive thresholding, and these two down here are just slightly different algorithm for that. One uses the mean for it, and one uses a Gaussian function to do the adaptive thresholding. And essentially that means as you're tracing along the dendrite, the minimum threshold that it's using can be modified and changed slightly so that anytime there's a dip in the intensity, which can happen, then it's going to allow you to still continue to trace, and it's not just this hole or a stop in the tracing or for the voxel scooping to happen. So that's how we deal with the uneven staining here, and Avia does allow you to kind of adjust this so that you can adjust, just a little bit of adaptive thresholding and a little bit of wiggle room, or a large amount of wiggle room, depending on what your dendrite signal looks like. All right. So I wanted to focus the workshop mostly in Avia Web and within the exercise so you all have hands-on experience. So let's get to the exercise now. And a couple of things just for Avia Web specifically. Jim is going to start sending out some links, and each of you will have a specific link to your own virtual machine for the web. Please only click on the link that is associated with your name. If you click on somebody else's link, you'll both be sharing the same virtual machine, and so you'll be kind of essentially fighting for control of it. So follow the link there, and it will basically come up with a splash screen for AWS App Stream. And go ahead and log in. Well, you might not have to log in. Sorry, this is only for me. You'll just be kind of launched into this DR Vision space here. Go ahead and click on Avia 882. CyberDuck is a way for us to kind of connect to other storage system if you're using your own data sets and things like that. For this, we'll be using the demo data set, so all you'll have to do is launch Avia. It's going to take a minute or two to kind of launch into this. So go ahead and start there, and then I'll kind of continue the presentation as this loads for everybody. All right, so I'm going to wait for everybody to kind of get in to finish up this Avia Web slide. So let me just go to some of the things that go over the exercise that we'll need. Okay, I see something in the...
Hey, Jim. Sorry.
Yep, go ahead, Chris.
Yep.
So there's a question I have. Maybe you're better equipped to answer. So one of the things we'd like to do is for our data sets, we're going to need to do a lot of manual validation. And one of the things that we're coming across is that our data sets are pretty big, even with cropping. And some of the open source options, like say YouTube, there's just memory limitations on that. Is there a way for us to somehow, with the license, try to use Avia Web so that we could distribute this out to, let's say, some of our student reconstructors, so they could do some of the reconstructions online?
So yes and no. The one big limitation of using Avia Web is the transfer rate. And you get a timed session. So when you are on Avia Web, you upload your images to a temporary folder, do all your work, and then download them. Working with them and sharing them and being able to visualize them would not be a problem. It's the uploading and downloading. But there's another solution that may work, which is we have what's called Avia Community, and it allows you to visualize images. So if your collaborators that are remote from the location where you are, if they have Avia Web, they can all be looking at the same thing, and you can all be doing screen sharing and looking at it that way. So that's kind of a way to get around that.
Well, no, it's actually that. So the way our pipeline we're trying to work in terms of our manual validation system is basically, we have our automated pipeline already, but we need to verify that. So we like to basically crop out some of our Imaris files and then, distribute it out to our students or other reconstructors, have them reconstructed and feed us back the reconstructed SWC or whatever format you guys use, the reconstructed files back to us. And again, the problem is that the open source software has limitations on memory, so some of our high quality, high res images have to be downsampled or cropped down really small. So I was just wondering if there is sort of a web-based option that could allow them to use Avia to do these limited reconstructions with our own data.
So with the way that Avia Web is set up in the current version here, that is not possible. Okay. Well, it's not that it's not possible, but kind of like what Jim says, you're going to have to deal with the data uploading and the data downloading. But we are kind of working on a beta version of Avia Web where you'll have your own virtual machine and it's constantly yours. And that basically storage that is permanent, essentially. That's kind of what you're looking for, right, Chris?
Yeah, that's basically what it comes down to. Yeah, if we could have our own private virtual machine to run Avia, that we could, give access to other people to hop on and hop off to use as they need, then that would, that might be a very useful solution for us.
Yep. So it's possible right now. It's kind of in its beta state. So we should probably talk offline about the details of that and getting potentially you up and running on that kind of system versus what we currently use for the web, the free version of the web, and also for the workshops.
Great.
Okay. All right. Okay. Any other questions? All right. Okay, that might have been sufficient time for everybody to get into Avia Web. Is anybody not into their Avia Web right now? Please raise your hand or message us or something to let me know. Okay, everybody good? All right, so just a couple of things here. So there's basically kind of two tiers, you can say. There is the file menu for Avia, which is kind of in the black screen, and then there's the file menu for AppStream itself. So that's the Avia, that's the AWS AppStream. So one of the things you might have to adjust is the resolution of your screen. So basically, to do some of the things we'll be doing, we need to see the entirety of Avia. So to check that, look at the bottom left of Avia. Let me get into my Avia here. And essentially what you're looking for is at the bottom left. If you don't see this little pencil icon, for the calibration, that means you're not seeing the entire screen. And on the very top right, you should at least see the analysis tool tab here. If you don't see those two things, you're gonna want to go into the cogwheel icon of the app stream and go to screen resolution and change it to about 1920 by 1080. That should give most people the ability to see the entire screen. You can play around with some of the other options, but that's probably the one that's going to work the best for everybody. Okay. All right. And just a note, depending on your internet speed and connection, you might get a bit of lag between what your mouse does and what Avia Web does. So just for probably more of the manual portion of it, just be wary of that and kind of have a little patience as we start to kind of manually trace and draw things. All right, just a quick snapshot of the anatomy of Avia so that we're all on the same screen. So down at the bottom here is the file gallery with measurements and spreadsheets and some editing and video animation. We won't cover video animation today, but we'll take a look at the spreadsheet. And then this big section is for data viewing and exploration. And then we have display setting. That's where you can control the intensity that's being displayed and also some of the cropping tools for Avia. And then this is the recipe console where we're probably spending the majority of our time, both with the recipe and with the manual tools. All right, so this is the exercise that we're going to do today, at least for this portion. So we're going to take a look at the 3D neuron analysis recipe, how to set that up, and this is the final result we're looking for. Basically, the tracing, the detection of the soma and the tracing of all of the dendrites that branch out of that. So this is the final results that we're looking for. Okay. All right, so everybody, let's get into Avia Web here. And we're going to load the demo data set. Basically, what you want to do is go to File, Open, and then here you're going to want to go to Share Data, Demo Data folder, and you're looking for Neuron Analysis Demo Data Set. Okay. So it's going to automatically load in the 2D main view. This bar, you can basically kind of scroll through to see the neuron. You can also adjust the intensity levels here. There's a little caret here if you want to pull that down and adjust those levels of intensity. You can also, of course, adjust the colors here. I'm going to probably do a green maybe, or let's do a slight red. So you can adjust those there. And then let's quickly launch into 3D so you can see the full neuron before we start tracing. These little dots, blue dots for each of these sections basically means that you can collapse this. So I just clicked on that to collapse it, and you can tilt and rotate the image here. Okay. All right, it allows the series of little blue dots also allow you to kind of adjust the size of these things. And these little pins right here basically allows you to pin these sections of Avia. So if I unpin this analysis tool, it basically collapses it down to here. And now this is one panel instead of two. I'm going to go ahead and put the pin back for that. Okay. So now let's actually go through the neuron recipe. So go to the recipe console tab. That should by default be the one that's already open for you. Take the drop down, push the drop down button, and then basically choose 3D neuron analysis. The recipes in Avia are organized such that they are application specific. So we're going to do 3D neurons. I'm going to show you this, although we're not going to really use it at this moment, but in this input and output section is going to be where you control what input channel you're working on. So all of the recipes are running on a single channel at a time. That's the structure that this is worked on for the majority of the case. The cell analysis can have multiple channels, but for the majority of the case, you're running a single analysis of a single channel at a time. So you have a multiple channel image data set, you're going to want to change this input channel. But again, it won't matter for us today. This is for 8.8 at least. This is also going to be where you find your region of interest. So I just turned on a region of interest, which pops up this kind of default rectangular box, and you basically move it and resize it using these little red dots. But we won't use that for now because this is a relatively small data set, so we won't really need to do the region of interest. The region of interest section, if anybody's working in Avia 9, is no longer in the input and output section. It's a little new section down right at the bottom of that. All right, so let's take a look at the recipe before we kind of delve into everything. So there's three main sections. There's the soma detection, there's the dendrite detection, and then there's spine detection. By default, spine detection is turned off, but you can always turn it back on by using these little red and green arrows, which are flexible pieces of the recipe that you can turn on and off. So here you can turn on the spine detection. Here you can turn on the soma detection. We have plenty of people who have data sets where they're focused on the dendrites and there's no soma in there. So they typically want to skip the soma detection. So let me just disable some of this. And then up here is where you control soma partition. So basically when you change these things, parameters will turn on and off. And you'll just kind of go in and fill it as needed. Okay, so let's set it back to skip soma partition and do soma detection, and then do not do spine detection. So that's kind of where we have it at right now. And that's what we'll need for the parameters for the analysis. So to set up the parameters here, it's easiest to go back to the 2D and do it in the 2D. So the first thing we're going to want to do is take a look at the range of soma diameter that we're looking for. If you already know what that number is, you can basically automatically input it into here. If you don't, however, you can always go to the annotation tool. I'm just going to delete the default here and basically measure it yourself. So I'm going to kind of scroll through the Z, find a good part of the soma to measure. And right now it's basically selecting the line drawing tool. And you can tell that by kind of the blue border that's around it. And you just basically click on one end to the other, and it will give you a couple of simple measurements. What we're looking for here is the length. And I'm wanting to take a look at the length of both kind of the shortest portion and the longest portion of the soma, so that I'm capturing the variety as much as I can. So the shortest looks about to be 11 and the longest looks to be about 17. So I'm going to go with 10 and about 18, just to give us a little bit of leeway here. And then the next is the dendrite detection. So the first one is dendrite diameter. So if you're detecting a soma, the best place to measure this is actually the dendrites that are directly coming out of the soma. That's going to be the best parameters to put in here. So again, I'm clicking from kind of one end to the other to get these measurements. Reading these length measurements, we're looking at 1.5 and 1. So let's say average is 1.25. If you're doing one without soma, I would suggest kind of an average dendrite diameter for this parameter. And then minimum branch length, basically the minimum length a dendrite branch can be. This, I basically, at the first iteration, kind of just measure what I feel like is decent and start from there, and then you can always adjust along the way. So this section I measured is just about 5. So I'm going to say it should be at least 7. And then the intensity threshold. So what you want to look for is kind of down at the bottom here, right next to where it says the 8-bit and all of the measurements. This is why we needed the entire view of Avia, because as I hover around the image, these measurements are going to pop up. It's going to pop up X, Y, Z in the intensity, which is the one that we want right now. So I'm going to zoom in and kind of take a look at the intensity for the background. I'm going to say about 20, which is looking pretty good there. And then the intensity for the dendrite. So this one, basically what you're telling the algorithm at this point is anything that is 20 and below, it's automatically background. And then for the upper threshold, it's basically anything above this is automatically dendrite or automatically part of the neuron. Everything in between, you kind of have a little bit of that wiggle room, which is where the mean intensity offset and that adaptive thresholding is. So that's the best way to think about it when you're setting these minimum and maximum thresholds. So I'm gonna set it at about 80. So this mean intensity offset is the adaptive thresholding piece. It's how wide you want it to be flexible. And this is in addition, this is going to be added into the intensity that you basically detect with that scoop. So I always like to start at 0, so no flexibility whatsoever. And then see what that looks like. And if you are missing parts of the dendrite, and especially the parts that are dark, then I would start to increase this a little bit. I would start at about 5-ish if you're looking at about an 8-bit image, and then about 500-ish if you're looking at a 16-bit image, and then adjust it from there. All right. How's everybody doing? We're good? Okay. All right, I'm going to launch back into 3D because all of the tracing is only, the results are only seen in 3D. And I'm going to kind of minimize that and adjust the view here. And then basically push start. And that's going to populate this object set setting. It's going to give you the soma, the dendrite, and the spine. So the object set setting is basically where all of your results are. It's where the mesh data is going to be. It's where you're going to be able to adjust the look of the result and also some of the turning things on and off when it comes to the visualization of the results. All right, everybody started their processing so far? We're doing okay in that regard? I'll also show you in a little bit, but there are different rendering modes here. And specifically for the dendrite. So you can have spheres, you can have surfaces, and you can have lines, things like that. All right, so that looks about done. So I'm just gonna pivot this around a little to kind of look at the tracing. So we did pretty good here. We're getting some of these dimmer dendrites out here, which is good. So that intensity level was able to capture those. So in Avia, basically as a general rule, these little eye icons control the visibility. So if you wanted to toggle the original channel on and off, you basically open and close the eyes. And the same with the neuron, and you can control each individual types of objects in the neuron with the soma and the dendrite. And the spines too, we had detected spines. So just a brief look here. I'm gonna turn this off. I'm also going to turn off this little box. So right where I am is in the 3D display option. I'm just going to take off the volume bounding box here so we can take a look at it a little bit cleaner. So right now we're in the sphere rendering of the dendrite set, and it kind of looks like what we're imagining with the voxel scooping, right? Scoops of ice cream. However, you can do it as a surface rendering, and you can also do it as a line rendering here. These little whites kind of dome and bubble here basically indicate where the dendrite is being connected to the soma. That's the point of connection here. So if we kind of take a look around, we can go 1, 2, 3, 4, 5, 6, roughly 6 points of dendrite connection between the dendrites and the soma here. You can also adjust the color of the meshes here. Right now it's color by cell. You can do a solid color and select whichever color you want to do. If I can just click on that. Yep, here we are. And that goes the same for the dendrite. So I'm going to basically have every dendrite segment at this point kind of its own color. So let me zoom out a little bit and so you can see that. Here. So then let me go to the spreadsheet. So down here at the bottom, because I've collapsed this area here. All right, looks like Chris. Can the reconstruction be exported? Yes, Chris. Here, let me show you that real quick. So File, Export, and you can export the Neuron objects. And basically, it exports as a SWC.
And Chris, we have somebody at USC who's doing a lot of this and then exporting it into YouTube. I heard you mentioned YouTube earlier, so I thought I would just bring that up.
All right. Okay, so we're in the spreadsheet. I'm going to change this to dendrite set because that's the one we have the most objects in. And these are the default measurements that we calculated for you and display for you. So we're looking at the path length, the mean diameter, the branch count, things like that. If you select on a dendrite, it turns white. And if you have a large data set and you have no idea where the selection is, this is a really nice functionality. Basically, zoom to selected object on the top section, top left of Avia here, and when you click on that, it will zoom to that selected object here. Okay, so we can go to another one and basically take a look at that. And there's a couple more measurements here. If these are not the measurements that you're looking for, if you click on this little ruler icon all the way to the left section here above the name of each of these segments, you'll get the measurement dialog. And here, if you go to soma and dendrite, there's a set of additional measurements that you can do, and you just basically have to select whichever one it is that you want and basically go for. Let me go to one for dendrite so you can see it. Let's do center line length and take some of these intensities away just to clean it up a little bit. And center line length. There's still a lot of it here. Center line length all the way at the end here. So you just need to select it and click finish, and it will create the measurements that you want here. Now, with these measurements, what you can also do is go back to the object set setting and basically now where you can color code by measurement here. And basically, once you get that, you can select whichever measurement it is that you wanted to do. Let's do average intensity. So there's a little bit of a range here. It's pulling it up right now. Let me see if there's a different one that perhaps might be. Let's do total path length. Yeah, let's do total path length. And then I'm just going to stretch this a little bit broader so you can see it. So now, and unselect this. So basically to unselect, just click Shift and then click on the object that you have selected. Am I clicking Shift? I might not be clicking Shift. There we go. So now the dendrites are color coded, and how did I set it? Blues are the shorter branch, and then as we get whiter and lighter, they're the longer branch lengths. So this is a really nice visual way to do the measurements instead of just the numbers itself. All right, so that's essentially all there is to the automatic recipe. The things that we went through and how we proceeded about this is, I think, just one kind of a starting point, essentially. And there is a lot of room and a lot of flexibility. So if you find that some of these are not correct, and especially this mean offset intensity, there's a lot of wiggle room here. So go ahead and, you know, feel free to kind of play around with those for your data sets. I find that the automatic tracing recipe works exceptionally well for data sets where you have a soma and dendrites to trace out. Even on larger data sets and a little bit more of a complex data set, if you have a soma to kind of a really good soma to have as a starting point for the tracing, this automatic recipe works really well. All right. So I'm going to pause there before we go into the manual tools. Any more questions at this point? Maria, if you have any questions, please feel free to kind of put them in the chat, or if you have a microphone, just speak them. I know you joined a little bit late, so some of the things I might be talking about, if you need me to repeat them, I'm happy to do so.
And Maria, I also just sent you a link if you'd like to participate in the hands-on portion of the workshop. Right now, Quinn is giving live demonstrations, and then everyone has access to Avia Web, where they can pull up the same image that she's showing on the screen and do the analysis yourself there on your local computer. So if you want to click on that link, if you have specific questions and want to chat me privately, I'll be happy to kind of walk you through getting to the point where she is now.
And Maria, I'm gonna give you a few minutes to kind of get that started, because it's going to take at least a minute or two for Avia to load. So I'm going to show some of the 3D display options to everybody as you kind of set up here. All right, so let's put the original data set back on, and let's take a look at some of these 3D display options here. So we have the clipping plane and we have an ortho slicer. So basically here with the clipping plane, let's put the bounding box back. Okay. So with the clipping plane here, you essentially have a clipping plane. You can change the axes pretty easily here if you want. So now we're clipping in the X or we're clipping in the Y. There's also an advanced functionality. Basically, it allows you to have full access to this widget so that you can create kind of crazier angles, not just things in the X and Y. So let's adjust this a little bit. To kind of adjust the where it is in the plane, it's these little arrows, but you can also type the positions in. So if you're looking for an exact spot, or there's a large data set that you want to see in a specific location, it might be easier just to kind of select this and put in the values here. You also have access to whether or not this clipping plane does the volume or the mesh. So we can basically have it just clip the mesh, and so you'll be able to see the full the full volume and just look at the mesh. This might be a good way to kind of adjust and examine how good the tracing is in, again, a large data set. All right. Let's go back to just being the Z slice. Okay, so here we go. This is a little bit easier for me to show you the adjustments. Okay, for the clipping planes, you can add about 6 different types of different clipping planes onto a data set. So that's again a really good way for you to kind of minimize that complexity when you're looking at a data set and trying to verify some of the data set. So it might be a really good idea for you, Chris, if you're going to try to kind of examine how good the tracing is on your entire data set. All right, so roughly kind of essentially the same sets of things for the ortho slicer, except now you're just getting a singular slice is the default. And again, kind of you can move it with the arrow, or you can adjust it in this bar too. And the X and Y also is, you also have that functionality and the volume in the mesh. Now, if we expand this caret down a little bit, it's going to tell you the position of the Z, but you also get access to this thickness. So by default, it's just 1 slice. But if you're looking and want to add a little bit more of that, you can always adjust that slice. So it becomes essentially a mini clipping plane or 2 clipping planes that are kind of in sync together for you here. So you can have a little bit more thickness if you need that for verification. So maybe a good point to do this is if you're verifying kind of a branch point, I find that you need a little bit more information to visualize both the dendrites that are being branched for verification. So basically, when you select it in the and it's in blue here, the planes are visible. If you want to, for visualization purposes, kind of have the slicer but not have the planes show up here, basically click on it again and it's still visible because the eye is open, but you can't visually see those red outlines anymore. So if you wanted to take a picture at this moment, that might be a little bit better to use. All right, I'm gonna go ahead and launch into the manual tools, unless Maria, if you're still having issues getting to Avia Web. Just quickly for Maria, the file we're looking at is if you go to File, Open, Share Data, go to the Demo Data folder, and then we're looking at the Neuron Analysis Demo .avia TIFF. If you can't find that, please message us on the chat and Jim will navigate you there. All right, so let's take these clipping planes off. I'm also going to turn off the automatic results and let's launch into the manual tools. So we're still going to be in the analysis tool tab, but we're going to be in a different sub tab. We're going to be in the 3D tools. So click on that. By default, it's probably going to pull you into a different tool. Yeah, Maria, the name of the file, yep, Jim's got you, Neuron Analysis Demo. All right, so it's probably in it's probably on a different tool, but what you're looking for here is the Neuron Composer, and I have it as the 5th icon from the top down. It's going to look like this. You might have to return those results that you did for the automatic recipe off again because it assumes that you want to adjust those. But we're going to start from scratch. All right. One of the things to kind of keep in mind as you're doing the manual tools is the manual tools basically assumes you as the user is the expert. So it's going to rely heavily on your expertise and also your adjustment of the intensity displays. So if you have something that's really super dim here and you want to trace it, I would suggest that you increase the intensity a little bit so that it's visible in the display here. And that's going to allow you to pick it up a little bit easier. So again, it's going to basically assume you have full control and knowledge of what should and should not be there. All right, so let's start with the soma. Let's add the soma in. So that's going to be in the edit mode, the last icon on the right. If you hover over it, it's going to say add soma. So click on that, it's going to select it, and again, that blue outline is going to show up so that you know it's selected. So there's a couple of parameters and adjustments here that you can do. There's a search range and then dendrite root radius. So we'll adjust that in a little bit. What I do want to make sure we all select at this moment is where it says add on, is to basically create a new object group. You can, of course, override what you did for the manual tracing, but for this workshop, let's create a new one so that we basically can do a comparison too. So let's leave it at that and then zoom into the soma and basically click on a location that's really bright, and you'll kind of get this gray surface that pops up, and that's where the soma is being detected. Okay, so I'm going to zoom out a little bit. Now, to kind of rotate the image and take a look around at all of the surfaces that this is detecting, make sure you hold down the shift as you click on the image and rotate. Because if you don't hold down shift, Avia is going to assume the click that you make is a new spot to start detecting the soma. Okay, so hold shift to kind of rotate and pivot the data. I think we're detecting too much of the dendrite, so let's decrease that search range a little bit. So as I move this, I'm going to move it a little bit dramatic so that you can see, but as it's basically looking at where you initially click out. So the higher this is, the more it's going to detect. I'm clicking shift, and I think this looks pretty decent. We're still detecting a bit of the soma, but that's okay, a bit of the dendrite, but that's okay. We can fix that a little bit with the dendrite root radius. Essentially, this is kind of the smoothing aspect of things. So if I bump this up to about 4, it's going to really smooth that out, and I anticipate that it's going to smooth out some of this dendrite sections too, because it's this tubular shape instead of, this roundish shape. So let's give that a go. Once you set everything up, click Create Soma, and it's going to take a bit of processing to create the soma, and here we are. So let me turn this on and off. So you can see we smoothed it out so that the majority of the dendrites was gone. You can go ahead if you wanted to remake a soma and get rid of this part, that would be okay. I'm going to continue on just for the sake of time here and assume that that's going to be okay. If we have time later on, I can also walk you through some of the cutting and editing tools for these meshes, and we might be able to solve it easier that way. Okay. All right, so we got the soma. Let's do the dendrites now. So the first one I'm going to show you is the predict dendrite. So that's the first icon all the way to the left. Again, there's a ton of adjustments here. First, we're going to look at the operations and making sure we're adding into the Neuron object set. This is the manual object set that we have, and not the automatic recipe. So I don't want to override that automatic recipe. All right, so basically how this is going to work is you provide Avia a starting point, and Avia is going to take a look at the surrounding pixels and show you a prediction of what it thinks the dendrite is going, and then you validate that. So because of that, and because especially when you're looking at large and complex data set, I find that most of them, the area around the soma is very dense, and it's sometimes a little bit difficult to find where the dendrite starting point is. I like to start at the edges of these dendrites. Those tend to be much easier for me to find in a dense data set. This one, of course, is not as dense, so you can basically start wherever you want. But I'm going to find kind of the end of the dendrite and start there. All right, so basically, I'm going to hover around the end of the dendrite, and you'll know that Avia has picked up the intensities there when the little ball that denotes your mouse is yellow. That means it's picked up some intensity and that you're good to go, essentially. So I'm going to click on that. And a couple of things to do, and this is a preference for me. You can go ahead and kind of decide whichever one you want to do, but I like the pan only, which means it's just going to slide the camera instead of rotating it around to get you the best capture of that section. But that's just a preference. And then the clip mode, I always want, I usually do latest. That's basically going to clip out all of the surrounding area and just show you the section that's closest to the prediction. Again, this is to reduce that complexity that we have in large data sets so you can solely focus on what it is that you're looking for. So this yellow is basically what Avia predicts the dendrite to be. And all we as the user has to do is select the right one. So that is, I think, trying to go into this dimmer portion, which I don't want to trace at this moment. So I'm going to select this yellow. And once you select it, it's basically going to predict the next section. And you can see here there's two predictions. It's thinking perhaps it's branching out here or it's continue along the trace. So I'm going to click that section, continue along the trace. And then the next section is predicted, and I just verify that, verify the next section. Let's zoom out and make sure this entire thing looks pretty good. I'm pivoting the data set around. That looks solid to me, so verify this last section. And I must have clicked on something wrong, but there we are. Okay, so I'm gonna unclip it and show you the full dendrite that I trace. You'll notice that it had automatically connect all of the segments that it predicted. It also connected to the soma and created that little transparent white bubble to indicate that it connected to the soma and not just to a different dendrite segment. Okay. All right. How's everybody doing? You guys predicting dendrites? We're good? All right. I don't see any questions. So I'm going to go ahead and do dendrite prediction on one more dendrite. And then I'm going to show you how to do this a little bit more manually. So I call this kind of a semi-manual method. I'm going to show you like the full manual method just in case you need that for some reason or this doesn't quite work out for you here. All right, let's go to this one. I'm going to go to this one because it's slightly dimmer than what we traced before. Again, I'm kind of starting on the edge here. Just to make sure this is a little bit easier, I'm going to bump this intensity up a bit so that it's pretty bright, or at least the starting point of this dendrite is pretty bright. All right, so again, let's zoom in so you see this a little bit better. Kind of hover over it. I'm making sure that that yellow ball pops up so that I know Avia detected it, and click to start. I'm going to go ahead and turn on that latest clipping mode there. And all right, so this is actually a really good section to kind of show you what we consider kind of the end-to-end prediction. So you can see here that this long line is mostly correct. However, it kind of pivots in the wrong direction. But since it's mostly right, I'm actually going to kind of basically do a slight adjustment. And what I'm going to do is I've dictated where the starting point is. I'm going to essentially dictate where the end point is. And Avia is going to trace as best as it can from the starting point to the end point. Can some form of adaptive thresholding be applied to the entire stack? So because the manual and semi-manual dendrite detection is based on the fact that you're the expert, there's not a ton of flexibility in terms of allowing you to do the adaptive thresholding. If you do have some data sets that are very tricky and the staining is really uneven, Chris, what I would actually suggest is we use the pixel classifier to enhance the dendrite and then trace based on that. I don't know if Jim's showed you that already. If he hasn't, and if we have time during this workshop, I can also go through that very quickly for you. Does that help answer your question? Okay, wonderful. All right, so I'm going to click pretty close to that area, and it kind of adjusted it and traced from end to end here, and that allowed me to get the correct tracing, but also not start all the way from the beginning. Okay, all right. So this looks pretty good. So I'm going to go ahead and keep clicking on it just to kind of show you what grow looks like. It's just slightly bigger. And then the full object set is basically the entire object that you're looking to trace. I'm going to go back to latest. Instead of grow, what you could also do is change this clip size. So just kind of change it along the way and adjust that. So you can also do it like that too. All right, so let's verify this next segment and keep going. And here it's not quite doing what I want to do. So we're going to do that end to end again. So I'm going to click on an area that's pretty close. I don't want to click on an area that's too far just because it might trace things that I don't want to. So I kind of want a little bit of a tighter control over this part. There we are. So just to kind of get it back on track, essentially, is how I like to think about it. All right, so we're heading to the soma now. We're at the end. So click on that. Let's remove this. So you can see here, it's also connecting and indicating that this is a connection to the soma. All right, so we got 2 dendrites traced here. Let's do one fully manual so that you have, you know how to use these tools yourself. All right. So I basically now have turned back on the original channel. And what I'm going to want to do is basically move from the predict dendrite to the dendrite pencil. Now the dendrite pencil, basically you draw the entire dendrite. So what you want to do, okay, let's select a short one. So for the first time we do this, so you all can see it. And I must also select a pretty bright one so it's easier to see on your screens here. So basically you zoom in, the dendrite is on. This trace diameter is basically the starting diameter that you want to kind of start and trace. I'm going to bump this up a little bit just for the fact that it's going to be easier for you to see where I'm tracing, but I have this selection of enhanced trace. What that's basically going to do is where I draw, it's going to adjust it's going to center it, and it's also going to adjust the diameter of the dendrite based on the actual intensity seen here, not fully on what I have input in the trace radius. If you don't want that to happen, you can just unclick this and it will just, those spheres and those diameter will purely be based on the value that you put here. Okay. So basically I'm holding down my left mouse and you can see the spheres kind of pop up as I trace, and I'm basically tracing it end to end. So once I let go and I turn this off, you can see that the dendrite is fully traced here and that the diameter, because I have the enhanced trace on, adjusts along the way. Now, because it's fully manual, it didn't automatically detect that this should be connected to the soma. We have to manually do that ourselves. And that's the next icon here, connect neurons. So we go there and basically we click on the left mouse and hold down the sphere that you want to connect. It's going to turn red and basically slide it almost to where you want to make that connection. And now you can see that this is that sphere, has that like white sphere that indicates it's connecting to the soma. Okay, I'm going to go ahead and repeat the same process with a longer dendrite. So just to quickly show you what dendrite segment to dendrite segment connection looks like. But the process is basically what we just did. Okay, so let's go to one that's kind of on the edge here. And I'm going to bump this intensity up so it's easier for you to see. We go back to the dendrite pencil. I'm going to draw one segment, leave it there. I'm going to draw another segment. Okay. I'm not 100% sure, Maria, what you mean by synapse buttons. Can you explain to me what that means and how that's typically seen in your image?
Hello.
Hi,
So
Maria.
I will. Hi. So what I mean, the buttons are the synapses. So the communication between the axon and the other dendrites, for example. Synapses can be axodendritic or other types. So they can be seen on the axons.
So let me just make sure I understand this correctly. So you're looking for a count on the connection between where the button is?
Yes, the number of buttons, because if the numbers of buttons are more, there is plasticity, and okay. So new connections was made.
Maria, is there normally you have a label, or is it morphologically visible?
No, it's morphologically visible.
So do you see one in this image?
So can you zoom out? Yes, if you zoom, so on the left there is a dendrite that is alone, let's say.
Maria, would it be easier if perhaps you share your screen and you can point it directly from your screen for us?
I don't have very much with me because this is something that was in a previous project. I don't have it. Maybe I can search on Google.
Yeah, or if you have Avia Web open with this image and you can see something on this image, you can just point to it, because it might. I don't know if we'll be able to figure out which one you're talking about there.
Okay, if you can give me some minutes to search something.
Okay. Yep. I'll do the tracing for this dendrite while you search for that, Maria. Okay. All right. So we have two dendrite segments here, and we're gonna need to connect them. So again, same tool, connect dendrite. and you basically click on one sphere and drag it to the sphere that you want to trace, you want to connect, sorry. And you can see it goes from red to green, basically, as I kind of move this away so you can see it a little bit better. And then once you make that connection, you'll see that there's a line essentially connecting from one section to the other. So you can do that along the way. And I stopped at 2 kind of short segments just for the purpose of this workshop, so you can see it a little bit easier. But as long as you can continue tracing, you can trace as long as of a segment as you want. There's not really too much stopping you, except perhaps some of the complexity that might be seen here in the data set, or if you have kind of sections where they're overlapping and crisscrossing each other a bit. Okay, so let's connect all of the sections that I've drawn. So again, one sphere one part to sphere another part, it turns green. And then this next section turned green. And here, if I take a look at the data set, these two are one dendrite that branched out. So let's make that connection too. All right. For this, when I connect it, you can see that the dendrite segment changed color. That's also another easy way for you to kind of see which dendrites go with which dendrite, basically, especially if I change this to color by label. So this segment is now its own color, this segment is its own color, and then this other branch is its own color. All right. Any questions? Does anybody want to share what their tracing results look like? I'm sure I trace different dendrites than you.
I'm not actually tracing anything, otherwise I would show you my traces. Let's see, but I might be able to find, let me see one that I did before. I might be able to show it here.
Let me know when you're ready, Luciano, and I can give you
Yeah, let me see.
green pairing.
8.
Oh, let me show you real quick, because people do ask sometimes how to delete some of these points. And that's the next icon here that looks like a trash can. So basically, let me turn off the original data. So basically, that's not a good one. Let's go to green so you can see the green color. So you hover over the sphere that you want to delete, and it turns a red color to indicate to you which sphere it is. And basically, you click on it, it deletes it. And we can delete a couple here just to kind of show you. And you can see the change in dendrite segments and color, so that to indicate to you it's a different part. And let me just connect those back again. So again, going to red to green, red to green, and now we're all in one single color again.
Okay, I have something I could show.
Okay.
While Maria is looking.
Examples for those boutons for us.
Yes. Good. So this one has a lot more cells, and it only has a small portion of it segmented. So here you can see this is actually I'm using Avia Community in this case. Can you? I guess you can see my screen now. So with Avia Community, this is good if you're if you have a collaborator and you've done some analysis already. So if you've done this analysis and you want them to have a look at it, you can send them the data set, like the data set you're analyzing right now. You can send them the data set and it will include all the results. So all of the results that you created, including the charts and the spreadsheet, all of that. And they can download this Avia Community from our website. It's a free download. Install it. They can open it up and explore it, and go look at the different dendrites, and make charts from this. Can do selections, all of that. So that's pretty useful. And because it's free, you can really give it to all your collaborators, so they can check out what results you've created. Also neat is you can make videos with this, like this little one I've just created. So you can create video animations from the results that you have generated. So I could do a new one here. So that's the full data set. Could start like that, then add these, come in. Then turn this off and look at the results.
Luciano, if I remember correctly, we did this analysis with the automatic recipe, correct?
Yes, this is done with the automatic one.
And I have something neat to show also. I think it's the same data set in Avia 9 with the transparency.
Oh, nice. Okay. Yeah, so that's all I wanted to show. So if you have created some results and you want to share with your collaborators or your friend or your mother even, as long as she has a computer to run, with the GPU, you want to show this is what I'm doing. And you can easily share it. That's what I'm talking about.
And we did mention Avia Community before also for doing collaborations with other people, because you can see all those different layers and results that it produces.
Yeah, great. Yeah, that's it. That's all I
just want to show this real fast to Chris. Let me see. So whoa, there.
Oh, it's blinking, Jim.
Yeah, I'm on a non-imaging computer, so this is, I don't know why. There, is it better now?
I am seeing a white screen
just
now.
White screen now.
Yeah.
Hang on one second. let me save it. Hang on. I don't know why.
Here's just posting the link, direct link to the Avia community page in case you want to download it right now, or forward it to somebody. That's the one that I was just using. The big difference is it's blue, first of all, not dark gray. So you'll notice straight away when you install it. And it has, it doesn't allow you to create any results, but it does allow you to create annotations in case you're training a machine learning classifier or pixel classifier. You can train a pixel classifier as a team and then apply it using the full Avia. Yeah, but it does allow a lot of stuff. There's a full list of things that it includes there.
So if you don't mind, I'd like to go back to that question that I think Maria asked about synapse counting. So
yeah,
can I show you the type of data that we'd love to be able to do very quick? I mean, I guess it's very close to what Jim is showing in terms of the reconstructions with this the spine counting. So our data is very similar. Do you mind if I share my screen real quick?
Yeah, go ahead.
And I have a picture up real fast, Chris. Mm-hmm. This is not Avia 9. This is still 882. It's on a different computer, but we can now make the different mesh layers transparent. So what I did here is I kept the soma. I'm sorry, I kept the spines the old way, but the dendrites and the soma I classified by color by class, and then made them transparent. So you can see it's a real neat effect. And it allows you, if you have intracellular components, you can see through the exterior inside of it. So I just wanted to show you guys that this is a feature in Avia 9.
And the spines on that one, those are automatically reconstructed?
Mm-hmm.
Yes.
Or generated?
That was all from the recipe. That was all actually in the same recipe that Luciano and Quinn were just mentioning, all the automated way.
So
Quinn, do we need to add? Oh no, it looks
I
like.
don't think so. No, yep.
Oh yeah, it does look like the other data set.
So can you see my screen?
Yeah.
Mm-hmm.
So this data is actually from our new the morph mice that I think I've spoken to Jim about. So the paper will be actually been accepted. So this will be coming out. So the mice will be available soon. But this is basically our sparse labeled labeling system in mice, and this is just basically clear tissue at 30x, and even at 30x we're getting pretty actually this might be 60x. Not, I think it's 30x. We're actually starting to see spines pretty well. So can we actually go in and count these, reconstruct or count these spines along the dendrites?
Yeah, can you zoom in? Like, so we see the details of a section where we can see the individual spines.
Yeah, I mean the individual sections look better. This is just the
MIP,
maximum intensity projection on my home one right now. I would have to dig through my workstation at work to get the actual stacks, because this is basically from my Maris, and this is just a max intensity projection. But yeah, but in the single Z stacks, the spines are much well resolved. Yeah,
that's what I was thinking. I mean, it looks like you will have really good resolution, right? It's going to look even better than what it looks like right now.
Yeah, it's extreme. It's reasonably, I would say it's reasonably sharp even at 30x.
Great.
So this is something that as we're doing the tracing, the skeletonization, can we go back and like 1 or 2 steps later and do the spine, have it reconstruct spines and just count it?
Mm.
Yep, that's for the recipe. We just kind of basically have to enact the spine detection portion, and part of the default recipes is part of the default measurements is counts. So that should not be a problem.
Should be able to see my screen now.
Yep, there we go.
And just so everyone knows, we technically have about 5 minutes left. So I just want to make sure if there are any last minute questions, we wrap up.
Yeah. Okay.
And get everything answered.
I'll show this quickly. So the spines are all counted. You can see them there. And it shows, in this case, 294 spines, and they are all listed here. You can create a little chart with any of the measured items. So the total spine volume versus, for example, total spine length, and that's all of them. We can also color code them by one of those measurements. So total spine length, for example.
And not to interrupt you, Luciano, but just to make sure that Chris knows as well, and Maria, you can also classify these. So I don't think I put that on the original quote to you, Chris, because it never came up, and it may not be something you need to do. But we have what's called an object classifier, which will now classify all the different spines.
Yeah, that's this here. So this is in case you have a group, like a phenotype of spines, and you want to separate them out, you can do this. So it will list all of them, show you a little thumbnail over here. You can then create as many classes as you want, maybe spiny,
length or morphology. And that one looks like it takes a 90-degree turn, and some of the others are fairly straight. So you can
mushroom. So you get the idea. You can create your own, and then you select them from here. Any one of them. You can select them from this thumbnail. You can select them from here, or you can select them directly from from here. And then you assign them to one of the classes. So I'm assigning these over here, oops, and a few others to the other class. Yeah, some measurements we were unable to reach all the measurements, but that's all right for now. So we have assigned a few examples to each class. You then teach, and then it will color code the spines when I apply. It will, hopefully it will do that. It says it's having some difficulties reaching some of the measurements for some reason. This is a very old file, that's why I'm not really concerned. So it might have measurements that don't work anymore with Avia 9. So once you apply, then it creates additional tabs down here for the different classes. So in this case, spiny and mushroom would be the two classes, and then these are color coded, and you can also study them separately in the charts, or you can color code them here as color by class. In this case, they're all the same class, so it doesn't really, nothing is going to change. But you can color code them by intensity. So spine, tortuosity. Yeah, and so you can also classify the dendrites and the somas. All of those are classifiable and color coded, which I think Quinn already showed earlier, so I won't labor that. Yeah, so the spines are just like any other object set. They appear in the spreadsheet, and you can color code them and interact with them in the charts like this. And that's it. All right, I will stop sharing my screen.
All right, just to kind of wrap it up, because I think we're running into that time. Maria, Chris, do you have any last minute questions?
For me, no. Thank you very much for this presentation and the answer for my question.
Oh, good. Okay. All right. I just wanted to quickly mention, since you signed up for this workshop, you probably already know, but there's a ton of other Avia online workshops that are going to occur in the next couple of weeks. Chris, I did mention the pixel classifier when you were talking about the adaptive thresholding for the manual input. So that might be another one that you would be interested to try, just to kind of enhance the neurons that you want